Okay, so the title for today is going to be Drug Discovery. And as the title suggests, the lesson is going to focus on how new drugs are discovered and then how those drugs are developed so they get to the point where they can be safely administered to people. So the first thing I'm going to jot down is that it is important to constantly produce new medicines. And the first reason why it's important to constantly produce new medicines is probably self-explanatory, but we'll jot it down anyway. So if I write down here why is it important that you are constantly producing new medicines? What we're seeing at the moment is that there is a new disease, COVID-19, that is causing huge problems in our society, and it's not something that we have any experience with. So we don't have any drugs for COVID-19, therefore it's important that we now try and make a new drug, okay? And COVID-19 is obviously a classic example of that, but there are always going to be new diseases that develop that we have to deal with. So it's important that we can constantly make new medicines. So under why, I'm going to write firstly, to treat new diseases. Okay, we haven't experienced every single disease and every single pathogen, so we constantly need to make new drugs to deal with the new diseases that we face, and COVID-19 would be a classic example of that. But there's another reason. Even if we had experienced every single disease and we had a drug to fight every single disease, we'd still need to make more. And the reason is what we talked about in the last lesson on antibiotics. So we learned in the last lesson that antibiotics are drugs that specifically target bacteria. And when we were looking at antibiotics, at the end of the lesson, I explained that some antibiotics can become some bacteria can become resistant to antibiotics. And what I mean by that is that the antibiotics stop killing the bacteria because the bacteria mutate, and they mutate in such a way that they are no longer affected by the antibiotics. Okay, they become a different form of themselves, and they're no longer affected by the antibiotics. And bacteria are mutating all the time. So because they're constantly becoming different forms of themselves, we constantly need to make new antibiotics so that we can always treat them. So as well as making drugs to treat new diseases, we need to be able to replace drugs that no longer work. As I said, the reason that those drugs will no longer work in this case is, bacteria can become resistant to antibiotics. And notice that I'm using that word resistant. Okay, we use that word when we're talking about bacteria being able to survive in the presence of antibiotics. We don't say bacteria are immune, we say bacteria are resistant. Okay, so we do need to constantly be developing new medicines and producing new drugs. But how exactly do you go about that? Okay, so when COVID-19 was first discovered and scientists set about trying to come up with some sort of treatment or some sort of vaccine, where do you even start? How do you start developing a new drug? And how do you go from the early stages when you're just trying to develop a drug to the point where you can safely administer it to millions of people? So we're going to look at drug development in a number of different stages. And the first thing we're going to do is we're just going to look at how you're going to identify the new medicine or the new drug in the first place. So where do scientists get their ideas for a new drug? And there's kind of two main approaches you can take. So I'm just talking about drugs in general. I'm not talking about COVID-19 anymore. So there's kind of two approaches that you can take when you're trying to identify a new medicine. You can study a specific disease. So if I said lung cancer, okay, you could study the disease lung cancer. You could find out what was going on in that disease. and then you could try and find a drug that would fix the problem. So you study the disease, you find out what's going wrong, and then you come up with some sort of chemical that might try and fix that problem. The other way of trying to identify a new medicine, and this is perhaps more common, is looking at plants and looking at microorganisms, because a lot of plants in particular have medicinal properties, so they have healing properties. And by studying different types of plants, you can isolate a specific part of the plant that has medicinal properties. So there's two different ways. You can look at a disease and try and find a drug that will match the disease, or else you isolate the drug and then you try and find different diseases that this drug will treat. So the first one we're going to write down is the one from the plants and animals, because plants and microorganisms, because that's very common. So I'm going to write extract. By extract, I just mean take the drug out of. So extract drugs from plants or microorganisms. Now, we've come across that word microorganism before. Micro means small, organism means living thing, because we saw that pathogens are small living things that cause disease. But this word microorganism just refers to anything that's small and anything that's living, so it doesn't have to be a pathogen. So extract drugs from plants or microorganisms that have medicinal properties. And this is how antibiotics were first discovered. Okay, so the very first antibiotic that was ever discovered was called penicillin, and it was discovered by a British guy named Alexander Fleming, and he discovered it completely by accident. He was growing bacteria on an agar dish, so I think I mentioned agar in the last lesson. We were talking about bacteria. So agar is just a jelly-like substance that bacteria can grow on, and he was growing some bacteria on this agar dish. And some mold started to grow on his dish, and where the mold grew, all of the bacteria died. And he realized that there was something in that mold that started to grow on his dish that was killing bacteria. And it was a type of mold called penicillin mold. And what they were able to do is they extracted penicillin from the mold, and they realized that penicillin was able to kill a wide range of bacteria. And then they took this penicillin, they put it into drug form so that you could treat bacterial diseases. So I'm going to write the antibiotic penicillin was extracted from mold. So it's one of the greatest scientific discoveries, and it happened completely by accident, where mold started to grow on his agar plate, and that mold happened to kill his bacteria. Okay, so the antibiotic penicillin was extracted from mold. Another classic example, okay, so that's an example. So mold would be a microorganism. So you're taking a drug basically that occurs naturally in a microorganism. Another example would be salicylic acid, which you probably haven't heard of, but the drug that you find salicylic acid in, aspirin, I'm sure most of you have heard of. So aspirin would be a common painkiller, and the actual kind of main key ingredient in the drug aspirin is called salicylic acid, and you actually get salicylic acid from the bark of the willow tree. And this was discovered a long time ago. The ancient Greeks knew that willow bark had medicinal properties. They had never extracted the active ingredient, but they knew it had medicinal properties. And then a long time later, scientists came along, examined the willow bark in more detail, and realized that it was this ingredient here, salicylic acid, that was causing the effect. And then they took this ingredient, they put it into the drug aspirin, and we now take it as painkillers. So, and there's so many other examples. Something like morphine, again, a really powerful painkiller that comes from the poppy plant. So if any of you ever watched Game of Thrones, they always talk about milk of the poppy, because you get morphine from the poppy plant, and it's a really powerful painkiller. So there's loads and loads and loads of examples of drugs just occurring naturally in plants and in microorganisms. And what scientists do is they study these plants and they study these microorganisms, and they take out the active ingredient. They isolate the active ingredient from the plant, and then they put that into drug form. So this is, you're kind of not looking at one specific disease, you're just looking at plants and at microorganisms and trying to extract anything that might be beneficial for your health. The other way to do it is this is a kind of more general approach. So you're trying to find the drug, and then you kind of look at the diseases you might use it on afterwards. And then the other approach would be to actually start with the disease and try and find a drug that's going to work on that disease. So I'm going to write down study diseases. So for example, at the moment, we've obviously got COVID-19. It's a massive problem. So scientists are specifically trying to treat the COVID-19 disease. And when you're specifically trying to come up with a drug for a disease, then you'll start by studying that disease. And by studying that disease, you'll typically start off in the lab. And to study it in the lab, you are going to grow cells. So what do I mean by that? So if, again, I'm going to use lung cancer. If I was studying lung cancer, if I was trying to come up with some sort of treatment for lung cancer, what I would do is I would get lung cancer cells and I would grow them in a petri dish in a lab, and I would continuously grow these lung cancer cells, and then I would be trying different things out on the lung cancer cells to learn more about them, like what do they need to grow, what kind of things will kill them, and try and learn and perform all sorts of different experiments, looking at their DNA, looking at how they metabolize, and form lots of different experiments to figure out as much as I can about these lung cancer cells. And then similarly with COVID-19, the first thing they needed to do was they needed to isolate the virus. They needed to learn more about the structure of the virus. And so I told you viruses have lots of different types of structures. They want to isolate it. They want to learn more about the virus, learn what the virus needs to survive. What does the virus need to grow? The more information you've got about the disease, the better you're going to be able to treat it and to come up with an effective treatment. So you're going to study the disease in the lab, but you can also use computer models. So once you've kind of got some basic information on the disease, you can use computer models, and you can use programs to build kind of like 3D molecules that mimic what you would have going on in your body. And then they have libraries with loads and loads of different chemicals in the libraries. And once you've kind of built your molecule, so with the COVID-19, they would have, isolated the virus, learned as much as they could about the structure and about what it needs to survive. And then they put that structure into a computer program, as much information as they had about it, they'd plug it into a computer program. And then you'll screen your structure of the virus against loads and loads and loads of different chemicals that we know about. And what the computer will do is it will bring up potential matches, like this chemical might work, this chemical might work, depending on the shape of your structure of the virus and then the shape of the different chemicals. They're going to try and find chemicals that will match the pathogen to see what might be effective on it. Now, this usually brings back tons and tons of hits. They call them hits. You could get thousands of hits, thousands of potential chemicals that might work on your particular molecule or might work on your particular disease. And then it's down to you in the lab to start screening and doing more and more rigorous testing until you can kind of isolate it down to maybe just one or two different chemicals that might have an effect on your disease. So it's quite a complicated process, and it takes a lot of time. There's a lot of trial and error with this process, starting with the disease and then working your way back to a drug. It takes a long, long time. So I'm just going to hear that you can search through libraries of chemicals to see which will act against your pathogen. Okay, and this is just a kind of a general statement. Okay, remember, not all diseases are caused by pathogens. So you're not always studying a pathogen and then trying to find a chemical that will, kill that pathogen. Okay, lots of diseases, cancer, heart disease, diabetes, none of those diseases are caused by pathogens. So when you're trying to find drugs to treat those diseases, you're going to use a slightly different approach. But in general, you will study the disease in the lab by growing cells, by using computer models, to find out as much about the disease as you can. And then once you kind of know a lot about the disease, you might come up with a target and then go through your libraries of chemicals to see if there's any available drugs that might act on your target and might help treat your disease. So just actually as an example, as I said, obviously cancer is not caused by pathogens. So what they did with cancer is you're not infected with anything. It's your own cells that just grow out of control. They grow way, way quicker than they should be. And because they're growing so quickly, you'll get tumours. So if you get lung cancer and the cells in your lungs are growing really quickly, much, much quicker than they should be, you can form like a big solid lump called a tumour on your lung, and that will stop your lung functioning properly. So they are your own cells and they grow out of control. So when scientists were studying cancer cells in the lab and they saw that these cells were growing much, much quicker than normal cells would grow in the lab. So what they decided to do was they decided to target the fact that the cells grow really quickly. So what they did was they came up with then chemicals that would only act, they screened their libraries to find chemicals that would only affect cells that were growing really, really quickly. Okay, so cancer cells grow really quickly, so we want drugs that only kill cells that are growing really, really quickly. And they were able to screen their chemical libraries to find drugs that would only affect cells that were growing really, really quickly. The downside of that then is it's not just cancer cells that grow really, really quickly. Okay, your hair grows quite quickly. And obviously one of the main side effects of chemotherapy is that your hair falls out, okay, because the cells that are involved in hair growth grow quickly, and they will also die when you're taking chemotherapy, which will cause your hair to fall out. So often you are obviously going to experience side effects when you're taking drugs. So when you're trying to come up with a target, by studying disease, you want that target to be as specific as possible to the disease so you don't get lots of unwanted side effects. But yeah, once you have identified your drug, identified your drug, there's a few things that your drug needs to be. Okay, so if I go back here to the antibiotic penicillin, I said you extract it from mold. Okay, so fantastic, molds can kill bacteria, but you're not just going to start giving people molds to eat to cure bacterial diseases. Same with, you're getting this from the willow bark. Yes, you can extract it from the willow bark, but then you need to be able to put it into drug form. So whatever these chemicals are that you extract from plants or microorganisms or whatever it is that you discover here from your computer models, you need to be able to put that chemical into drug form. And drugs need to have the following properties. So all drugs need to fit certain criteria, or they need to be checked for. They need to be checked for their safety. Okay, that one's the obvious one. You need to make sure that the drug isn't going to have lots of unwanted side effects. Now, I mentioned with chemotherapy, a side effect is that you hair loss. Obviously, that is not a desirable side effect, but it's not going to kill you. Losing your hair, as awful as it might be, it's not going to kill you. And so when we're talking about safety, we're talking about really bad side effects that will affect your overall health. So drugs need to be safe. They also need to be checked for efficacy. And what I mean by efficacy is the drug effective? Does it do what it's supposed to do? So if you're taking a painkiller, or if you're trying to make a new painkiller, well, then that drug has to kill pain. Okay, that's the point of the drug. It needs to be effective. So we say we test for efficacy to make sure that the drug can do the job that it is supposed to do. And then the last thing drugs need to be checked for is dosage. And by dosage, I just mean how much of the drug is appropriate. And the dosage is a bit of a balancing act between the other two, because too much of the drug and it might be unsafe, it could cause unwanted side effects, but too little of the drug and the drug might not be effective, it might not be doing what it is you want it to do. So dosage is really important. How much of the active ingredient are you going to put in your drug? Okay, so for example, when you take your aspirin tablet, I said the active ingredient in aspirin was salicylic acid, but it's not the only thing that's in that little white tablet, okay? And the amount of the active ingredient, the amount of salicylic acid that you're putting into your tablet, that's going to be the dosage. If you put too much salicylic acid into your little tablet, then it might be unsafe. If you put too little salicylic acid into your little white tablet, then the drug might not be effective. So dosage is really important to get right. So in order to test drugs properly, to make sure that they're safe, to make sure they work, to make sure the dose is correct, you need to go through 3 different sets of tests. Let's go to the other page. So there are 3 stages of drug testing. Three stages of drug testing. And stage number 1 are the preclinical trials. Okay, so preclinical, clinical refers to anything that's actually with it in the hospital, and this is pre, so it's before. So it's before the hospital, before it's being given to people. What exactly is done? And the first thing that you're going to do is you're going to test the drugs on cells. Cells that are just grown in the lab. So again, if I was trying to come up with a cure or a treatment for lung cancer, I would start by just growing lung cancer cells in a dish. And then I would take the drug that I wanted to test and I would put it on the cells in the dish and I would see what happens. Okay. And what I'm looking for is I'm looking for the drug to be safe. Okay. So I don't want all of the cells to just immediately die. I don't want it to be really toxic. I do need it to work. So whatever the drug is supposed to do, I need to see that happening. And then I'll also play around with dosage and see a rough idea of how much of the drug is needed. And many drugs will fail this test. Okay, most drugs, majority of drugs that you're testing don't go past the preclinical trial. They don't move past cells, either because they don't work well enough or they don't do what they're supposed to do. So in a cancer drug, you might just want the drug to make the cells stop growing. That could be the aim. Don't kill them, just make them stop growing. If you're putting your drug on and it's just killing them instantly, then it looks like that drug might be too toxic, so you would just abandon it. On the flip side, if you were putting quite high dosage on and literally nothing was happening whatsoever, well, then you would say that the efficacy was poor, that the drug wasn't effective, and it would fail the trial. Okay, so preclinical trials on cells, that's going to be step 1. If it passes preclinical trials on cells, then you are going to move on to animal testing. Animal testing is what it says it is. Drugs are tested on animals, and that is usually mice. Not always, it will depend on the disease, but mice. It will usually be mice. So obviously animal testing is quite controversial. A lot of people would feel that it is wrong to just test these drugs on animals. It is necessary. I do believe it is necessary. No matter how successful your preclinical trials are, and you can use cells and you can use computer models, there really is no way of predicting how it's going to affect an entire organism until you test it. So when I'm testing my lung cancer drug, I'm only testing on lung cancer cells in a dish. I have no idea how that drug is going to affect the liver or the brain or any of the rest of it. I need to test this drug out on a full system. Okay. I also need to make sure that the drug can get to the organ that I want to get it to. So typically you're giving a drug, you're going to take it in tablet form, or you're going to inject it. The drug will first go into the bloodstream and then it will travel all around the bloodstream. In order to check that this process works correctly, you need an animal model. You can't get that from cells. So drugs, they are tested on animals. Now it's not, in this country anyway, it is illegal to test cosmetics on animals, okay? And it's illegal to test any tobacco products on animals, but all drugs that are going to be used on humans for medicinal purposes all have to be tested on animals. So it's a required stage. But as I said before, this stage here has become a lot more stringent. You have to go through a lot of procedures in order to pass this stage. And most drugs do fail here. It's only the drugs that are very, very promising that will then move on to animal testing. And I'm not going to lie and say animal testing is, you wouldn't want to be a lab mouse, like you wouldn't, to be fair. Because they will take the drugs and they will inject it into the mice, and they will monitor the mice, and they will see how the drug affects them. Ultimately, the mice will all be killed. They'll be sacrificed, because at the end of the experiment, you're going to want to do an autopsy. You're going to want to have a look inside and see how the drug has affected the different organs of the mice. So ultimately, they will all end up being sacrificed. But yeah, animal testing is a necessary part of the drug development process. It's usually done on mice, but other organisms can also be used. Okay, so once you have passed the animal tests, then you move on to what we call the clinical trials. So clinical is when you're actually in the hospital setting, human clinical trials. And you first start off with a very small number. Okay, so you've never tried this drug in a human before. The last thing you want to do is give it to lots of people at once. You'll start off with a small number, and they'll be healthy volunteers. Okay, they will not have the disease that you're trying to treat. They will be entirely healthy volunteers. And the reason being is at this point, you're not looking to see if the drug works, you're just making sure that it's safe. And it's less likely to have ill effects on someone who's completely healthy than someone who might already have a compromised immune system or be struggling with another disease. So you'll take a small number, and they will be healthy, and they will have volunteered, and they will typically get paid to volunteer in this study, okay? Because they've got nothing else to gain from it other than the fact they might get some money. They're not sick themselves. They just put themselves forward. So you get a small number of healthy volunteers. If that passes, then you're going to try on people with the illness. Move on to people with the illness. So in this step here, you're testing more for the drug safety. Is the drug safe? When you're looking at the people with the illness, now you're checking for efficacy as well. You're wondering if the drug does what it is supposed to do. You are initially going to start off with low doses. So we're talking about dosage. You're going to have low doses. They're going to be initially used to make it as safe as possible. But then you are going to increase the doses until you get to the optimal dose. You'll start off small, and then you'll increase it. But these trials are never risk-free, okay? Even coming from preclinical trials into animal testing, into clinical trials, and these tests are never risk-free. You're always entering into a clinical trial at your own risk. And there are, because I know we're talking a lot about the coronavirus and why does it take so long to get a vaccine and all the rest of it. These steps are incredibly important, and skipping a step or moving from one step to the other too quickly can have really, really negative health benefits. So, for example, there was a study that was done. It wasn't even a study. It was back in the 1950s. There was a drug that was developed by a German company, and it was developed initially as a sleeping tablet to help you sleep. And it also had other beneficial effects. It was really good for treating nausea. So although it was initially prescribed for people who had trouble sleeping, because it was working on nausea, lots of pregnant mothers started taking this drug to help treat morning sickness. And the drug itself had been through all of these tests, and there was no kind of toxic effect at all. They never had problems with safety or toxicity. So lots of pregnant mothers were taking this drug, and what they hadn't looked at any point of the trials was the effect that the drug would have on an unborn fetus. So while the mother had no ill effects of this drug, unbeknownst to mothers, the drug was having a terrible effect on their unborn fetus. And there was a lot of babies then born that were missing limbs. So they either were missing limbs completely, so missing their arms or missing their legs, or just their arms or legs just hadn't developed properly, and because this drug was affecting that stage of development. And it took a long time for them to put kind of 2 and 2 together, because it took them a long time to link this drug to these deformities. And this drug was being taken for around 10 years. And overall, they think after they found out that it was this drug that was causing all of these abnormalities, they think by the time they figured it out, over 10,000 children have been born with some sort of deformities. So it's really, really important that these tests are done properly and done thoroughly, and that every single aspect of toxicity is considered. So something like the coronavirus vaccine, they could have this in trials and they're taking it and there's no kind of toxic effects, but some toxic effects are long-term, they're not short-term, and it will take a while before you see them. So it's important that you wait long enough to kind of look at all the effects that this vaccine might have had in the person before you say that it's definitely okay and that it's definitely safe. That drug I mentioned, that was back in the 1950s and 1960s, but far more recently, in 2006, there was a clinical trial in London, kind of a high-profile clinical trial for a new drug to treat leukemia, which is a type of blood cancer, and they had 6 men who were all injected with the drug, and within hours, the men were all rushed into hospital. They were vomiting. They went into organ failure, and it affected the circulation. So some of them had to have some of them just had their toes and fingertips amputated. Others had to have their whole hands amputated. One guy's head just swelled up completely. It's known as the case of the elephant men, because his head swelled up, and apparently, according to his girlfriend, he looked like an elephant. But it's a case that which received a lot of publicity, because this was done, kind of recently enough in London, and all the tests were supposed to have been done and have been carried out properly. Yet these 6 volunteers went into organ failure almost immediately after taking the drug. So it's really important not to rush these things, and make sure that it really is 100% safe before you start making it available to the public. I rambled on for a lot longer than I normally ramble on. It's because I figured out how to upload videos longer than 15 minutes, so I'm not kind of bound by my restrictions. I promise I'm going to stop talking now. I'm just going to put up a question. It's a 6-mark question on drug disease. I want you to have a go at answering it, and then I'm going to answer it with you, and then that is it. So just one 6-mark question to summarize everything that we have done today, and then we will leave it at that, and I promise I'll stop talking. So, you can't see that at all. Boom, there you go. So it says, antibiotics are drugs that treat bacterial diseases. The development of a new antibiotic involves several stages of testing before it can be prescribed. Explain each of the stages in the development. So 6 marks, there's 3 different stages that I've just talked through. Explain each stage, saying what the stage is and why it is important. Have a go by yourself, and then I'm going to go through the answer with you. So press pause and then press play when you're ready to see the answer. Okay, I've scribbled it down and answer. Okay, so you've got 3 stages that you need to talk about. Preclinical, so it's before the clinic, so that's when you're still in the lab and you're testing the drug on cells. First of all, just to see if it's effective, does it do what you want it to do, and is it safe? Okay. You're also going to test the drugs on animals. This also counts as preclinical, but it's a different stage of preclinical. You test them on animals. You want to find out the effect of the drug on the whole organism. So when you're just using cells, it's just one isolated organ system. One isolated tissue. You need to move into animals to check out the effects on the organism as a whole. So it's important to kind of make that distinction. That's why animal models are much better than just cells. And it can also help you figure out the right dosage. So when you're giving a drug to cells, if you just got growing in a dish, all of that drug goes directly to the cells. When you've got an animal and you're injecting the drug into the bloodstream, that drug is going to go all over the animal. And so it might not all go to the target area, so you might need to give a significantly higher dosage within an animal model than what you had to give to your cells. So when you're looking at just your cells, it's hard to kind of estimate any kind of dosage. when you're using your animal model, that's when you're going to start looking at dosage. So it's important to mention dosage in your animal model, and also that you're looking at the effects on the whole organism. Then you're going to go to clinical testing. So preclinical is the cells and the animal models. Then you move into clinical. That's when you start to give it to people. You're first going to give it to a small number of healthy volunteers, okay? Because these volunteers are healthy, you are not looking at the effectiveness of the drug. These volunteers do not have the disease that you are trying to treat. Therefore, you cannot see if the drug is actually effective. What you can see is if the drug is safe, okay? It will also help you to check for your dosage as well, see what dosage works best. So you'll typically start with a smaller dosage and then you'll increase, just to the point that they might start to experience side effects. Once you've gotten the all clear from healthy volunteers, that's when you're going to move it on to people with the actual disease. Once you're treating people with the disease, that's when you're going to see how well it works. So you're not going to be talking about the effectiveness of the drug in healthy people, but you will talk about how effective the drug was when you're testing people with the disease. Again, you're also going to look for side effects, okay? And you're going to do with this, here was only a small number. The second trial is going to be a much, much larger number of people. Different people react differently to different things, so it's important to have quite a large sample size, just to see all the potential side effects, how common those side effects are there any kind of pre-mitigating factors that might lead someone to get a side effect while someone else doesn't. You want to get as much information as possible, and these trials will last quite a long time. So it typically takes a few years for a drug to get from the stage where you're just doing preclinical tests to the point where it's actually available to the public. Obviously, something like COVID-19, where they're desperate for a vaccine, they might kind of speed up this process a little bit, but it's always going to take time to get from one stage to the other and make sure that you are doing your checks properly. Sorry, this video has gotten ridiculously long. Apologies.